Here are the answers to the critical thinking practice questions for Chapter 10. Here's our first scenario. We've got a patient with pneumonia, and we do a gram stain, and we find that the cells stain purple, and they're circular, and they are in a chain arrangement. So for the next question, I'm asking you what kind of drugs would be best to use for this patient. So you have to know from the initial scenario what kind of pathogen it is. And the part that gives you that clue is that they stain purple, they're circular, and they're in a chain arrangement, which means they're gram-positive streptococci. So going back to the first question, which of these drugs are effective against bacteria, specifically against gram-positive bacteria? Well, gram-positive bacteria, unless they're antibiotic resistant, tend to be more susceptible to antibiotics than gram-negatives. So everything that will attack a bacterium would be effective. So it would be all of these guys. And yes, I got ampicillin twice. I'm going to have to fix that. Now, I like asking this type of question on the test, except it'll be easier on the test, because I'll only have one antibiotic that's effective against bacteria. I'll have one antifungal. So suppose the patient came in with pneumonia symptoms, but it's a eukaryotic cell with a cell wall that indicates that it's chitin. Then you would look for, say, amphotericin B to treat this patient. Let me know if you have questions on this type of exam, especially since on the exam I won't be asking you to know any drug that's not on the worksheet. So make sure you know the antimicrobials that are on that worksheet that I had you do for this chapter. Next up, from this same initial scenario, in the lab, a test for drug susceptibility was also performed. Which of the following was likely performed? Now, all of these are tests or refer to antibiotic susceptibility, except for the allergy scratch test, so you can ignore that one. Now the question is, what would be done in a clinical lab? Well, the disc diffusion test, or the Kirby-Bauer test, is definitely done in hospital labs. The E-test is done occasionally. Minimum inhibitory concentration is done in research labs when they're figuring out therapeutic index. So that one would not be done in a hospital lab. Therapeutic index, also done in research labs, so the answer would be B and occasionally C. Now this next scenario, here we have extracts from Samoan plants, and we show minimum inhibitory concentrations for these potential pathogens. We have 2 gram-negatives, E. coli and Pseudomonas aeruginosa. We always throw Pseudomonas in because it is naturally resistant to most antimicrobials. We throw in an acid-fast bacterium, we throw in a gram-positive, and we throw in a yeast. Now down here we have human fetal lung cells. These are normal human cells that are grown in culture. They're grown separately from a human body. We also have human cancer cells that are also grown outside of the human body, but these are cancer cells. Now, the next thing that I need to show you on this chart is just a reminder, micrograms are bigger than picograms. Picograms on the metric scale are 10 to the minus 9th of a gram. Micrograms are 10 to the minus 6th of a gram. So micrograms are about 10 times bigger than a picogram. Now, so this is the minimum inhibitory concentration. This is the amount of the extract 498-WP, that was effective in having the tube of these pathogens completely clear. Now, over here, we've got, instead of an MIC, 50% kill of the cultured. So let's go to our first question. Why did we include the human cell lines? That's a good question. Are we looking to determine the toxicity levels of the extract? Maybe, since they're both human cell lines, and we're looking at how the extract would behave with humans. Now, it's not in a human body because these are individual cells. So, that's a possibility. As a eukaryotic negative control, well, that's a good idea, except we included yeast. So we've already got a eukaryote in there. To see if the extracts have anti-cancer activity, well, we did include a cancer cell line. That's a possibility. To determine the cancer-causing potential of the extract. Okay, that sounds really good. But think of this. Cancer shows up in multicellular organisms. And generally speaking, when we do this kind of testing, our cell lines don't generally become cancers. So we're going to eliminate that one right there. We're going to eliminate this one. So there's that one. We're looking to determine toxicity levels. How does it behave with human cells? And that would include our normal cell line, and that would also include our cancer cell line. Then for C, that's a possibility, but just for the HeLa cells. We're not looking for anti-cancer effects on our normal cells. Okay, for our next question relating to this scenario, extract 498-WP would be best looked at for development against which of the following types of pathogens? So we have acid-fast, cancer, fungi, gram-negative, gram-positive, and lung cancer cells. Well, let's go back and look at the table. Okay, so let's look at what it was effective against. So, you know what? I'm going to take a look at my human cell lines first because I want to look at toxicity. So, for the normal lung cells, we had 50% of the culture cells killed at 75 micrograms. So we want to stay away from 75 micrograms because you don't want to kill 50% of your cells in your body. Actually, that wouldn't happen. But it's a good basis to go with. Now, for the human cell lines, the human cancer cell lines, it only took 150 picograms. Yeah, that's interesting. So, do we want to consider the fact that it kills cancer? Let's keep that in mind. So, let's look at what is below the range of 75 micrograms. So, for E. coli, it's 150. That's twice the toxicity level. So, we wouldn't look at it for E. coli. Nor would we look at it for Pseudomonas, so the gram negatives are out. This one's out. The acid fast is out. But here we have 25 picograms for gram-positive and 15 picograms, so it did even better on the Candida. And then we have 150 picograms for the cancer. So, it looks like we can eliminate A, B is a possibility, C is a possibility. We're going to get rid of D because it's not gram-negative bacteria. Gram-positive bacteria, lung cancer cells, that would be all fine and good, but this is more specific than this. I think we'd want to go with a broader range there. So our answers would be cancer, fungi, and gram-positive bacteria. Okay, now for our next question, what is the likely cellular target of the extract? Here we're trying to combine what is similar between cancer cells, yeast cells, which are eukaryotic, and gram-positive bacteria. Now, for this one, I don't really know the answer. I pulled this information from an actual test, and I wasn't around when they figured out what was going on. But one thing to keep in mind when you're talking about plant extracts, there may be several chemicals that are doing several different things. So it's okay if they're widely different. But the best approach to take a look at this is to look for similarities. So what is similar between yeast, gram-positive bacteria, and cancer cells, human cancer cells? Well, both yeast and gram-positive bacteria have a cell wall. Well, gram-negative bacteria have a cell wall, and it's made of peptidoglycan like the gram-positives, whereas the yeast cells are made out of chitin. So is there a similarity between the cell walls of yeast and gram-positive bacteria that doesn't relate to what it's made of? And the answer is yes. They both have a thick layer of whatever the cell walls are made out of. Whereas gram-negative cells have that outer membrane. They have almost like a lipid bilayer on the outside of the cell. So, they're sort of similar there. Would we want to say prokaryotic cell synthesis? Well, that would be grouping gram-positives and gram-negatives. And we can see that the gram-negatives are doing better against this extract than the gram-positives are. Would we want to go with prokaryotic protein synthesis? That's a possibility. But once again, it was attacking gram-positives versus gram-negatives. Therefore, I would think that because they have the same kind of ribosomes, 70S ribosomes, probably not. So would it attack both eukaryotic and prokaryotic protein synthesis? Yeah, but I'd still have a problem with the prokaryotic protein synthesis. Fungal cell membranes. That's a possibility. Okay, that's a possibility. Because remember, we can have different things in the extract doing different things. In which case, we could put prokaryotic cell wall synthesis back on the list. Because gram-positive cells are more sensitive to antimicrobials that attack cell wall synthesis than gram-negatives are, because they've got more cell wall. And then rapid mammalian cell growth. Sure, why not? The test indicated that it was effective against human cancer cells. They're growing uncontrollably. Yeah, why not? So I would say the most likely answers would be definitely fungal cell membranes, rapid mammalian growth. I'm still not sure about the cell wall synthesis. I'll put a maybe on both of them. I'm very not sure about the protein synthesis. But if you came up with a good argument, sure, I'd take it. Next up, if you decide to develop this extract for use against gram-negative bacteria, what would you need to keep an eye on? Well, definitely allergic reactions. So as you go on testing, you want to make sure that we don't get a whole lot of that. Antagonism with other microbials? Yep, you want to make sure that if you use this as a treatment, if it does have antagonism or it doesn't play well with other antimicrobials, you don't want to use them at the same time. Therapeutic effect. Yes, you do want to keep an eye on that, but that's combined in therapeutic index. You take the therapeutic effect, which is your MIC basically in this table, and divide that by your toxicity or toxic index to get your therapeutic index. So I would say you definitely want to keep an eye on the therapeutic index and the allergic reactions and antagonism. Well, that's it for this critical thinking practice. Hopefully it was useful, and it'll be useful to you in studying for the exam.